great panel, great set of panelists here, and me. And our guest group have done so much work in neurodegeneration, neuroinflammation, the level of research and drug discovery. And so earlier today, you heard Ann Klibanski tell you that one of the main emerging themes from the last forum was the merging of neurodegeneration and neuroinflammation. And in Alzheimer's disease, you know, at the neurodegenerative side, we have amyloid plaques, we have tangles, and we also have neuroinflammation. And there's been a lot of work done in terms of which pathology comes first, which pathology is absolutely necessary for seeing symptoms of dementia. There's been work on resilient brains from Teresa Gomez-Isler at Mass General showing that you can have a head full of plaques and tangles, and if you don't have gliosis and inflammation, you can deal with plaques and tangles for quite a long time. So I'm going to start by just introducing panelists. I think you saw them up on the screen, but we have Mike Hutton from Lilly and Roger Nitsch from Neurimmune, Hugh Perry from University of Southampton, and Christian Haass from University of Munich. Is that still the name of the university? Because I know you, a different name. And Michael Hayden from Teva. So we're going to be covering all three aspects of Alzheimer's pathology, also cross-cutting into other diseases. And I want to start with a question to Mike. So is amyloid no longer a good target for Alzheimer's disease? I need to get them riled up.
So I think amyloid remains a very important target for Alzheimer's disease. I think we need to look at other areas, but I think it's still a very important target. The genetic evidence suggesting that amyloid is a key player in the disease, an early initiator of the disease process, remains absolutely compelling, not just from the rare variants that impact early onset disease, but also the protective variant that we see in the Icelandic population, so mutations that both increase risk and decrease risk, all consistently affect A-beta. And also ApoE4, of course, which is the most common risk factor for idiopathic Alzheimer's disease. We don't understand exactly what ApoE4 does, but we understand for sure that it impacts amyloid deposition. So let me turn to the trials, and obviously, you know, we were very disappointed at Lilly to see the results of solanezumab and, of course, the results from the Merck BACE inhibitor. I think, having said that, though, it's difficult to say what solanezumab has really told us about the role of A-beta in the disease, and there's two reasons for that. Firstly, although we missed our primary endpoint in the EXPEDITION3 trial, we do think that there's a small clinical signal there, about 10 to 15% shift in the rate of progression on each of the cognitive and functional endpoints that we looked at, and many of those actually were statistically significant. So I don't think we can just dismiss that clinical signal and therefore the effect on A-beta that we see with solanezumab. And then the other key point about solanezumab is that the target engagement is not that high. About a 15% reduction in CSF free A-beta. So I think that's another key point to make. Right. The Merck BACE outcome, I think, is potentially more problematic. Those patients, we would expect to see a 70 to 80% reduction in A-beta production, although we wait to see the results. I think interpreting exactly what they've seen, of course, we have to wait for the results to come out. That's something that we await with great interest. So overall, in terms of what we need to do for A-beta at this stage, I think there's three things. I think the importance of maximizing target engagement. I think the Merck results probably tell us, although again it's too early to be really sure, that we have to go early in the disease process, particularly for therapeutics that are targeting soluble A-beta. And I think the other point that Roger will come on and address in a second is, are we targeting the right species? So do we need to move to targeting the insoluble amyloid plaques through plaque-specific antibodies such as aducanumab, or potentially do we need combination therapies that target both BACE and both soluble A-beta and the plaque A-beta? We've looked at Lilly at combinations of a BACE inhibitor with a plaque-specific antibody, and those really do give us very, very impressive results. And then potentially long-term, looking to amyloid and tau type of combinations.
Yeah, before I get to Roger, so, you know, immunotherapies clearing the amyloid out, BACE inhibitors and gamma secretase modulators are lowering the production of A-beta. Is there any evidence that if you just lower production of A-beta, that you'll start to clear amyloid out of the brain as well?
I think when we've done it in transgenic models, if you hit A beta production high enough, so if we reduce A beta levels sort of 70, 80, 90%, then that's the type of equilibrium which will, at least in the amyloid mice,
When they already have amyloid, though?
when they only have amyloid, when that will produce a reduction in sort of cord amyloid load. But inevitably, I think the effect on amyloid through just simply lowering soluble is going to be limited. If you want a robust effect on amyloid in the brain, such as we've seen with aducanumab, you're going to need probably a different approach, plaque-specific antibody being the most obvious.
And just so you know, I'm not tweeting or texting. I have my notes on my phone. So Roger, tell us the Ajik Canyon story. It's fascinating. You know, this was a very different approach to immunotherapy. It came from learning from natural mechanisms in humans, and maybe that will explain a little bit about why Ajik Canyon has so far looked so much more successful as an immunotherapy.
Well, aducanumab is a different molecule, first of all. It does not bind to soluble A beta, does not bind to APP, but it has a very selective affinity to all kinds of aggregates, soluble A beta aggregates, and also fibrils, and also amyloid plaques. It migrates over the blood-brain barrier into the brain, binds there, activates microglial cells that then will phagocytose the material. And the effect on amyloid removal is both time-dependent and dose-dependent. So the higher you go in dose, or the longer you treat, the larger the effect. So in the highest dose that we did in this trial, 10 milligrams per kilogram, after about a year or so, there are patients who are actually down to the level of amyloid that was required to be included in the trial. So that would probably tell us that it's almost complete removal of beta amyloid within 1 year. If you go longer with the doses, then that effect will continue also into the second year. So even though this was not a cognitive study, and the study was way too small to do formal cognition testing, which we are doing now in the phase 3 trials, when we separated patients with a high degree of amyloid removal versus a low degree of amyloid removal, it was very clear that the ones with high amyloid removal were cognitively stable, even though it was a small trial, and this is a post-hoc study that's probably teaching us that it's really important to remove amyloid. So maybe I can just add one more point. The BACE inhibitors, I think, would be a great idea to continue the treatment after amyloid removal with an antibody to prevent further buildup.
So if someone has imaging and these drugs are online and they have a lot of amyloid in their brain, you might suck it out with aducanumab and then maintain with a BACE inhibitor or a GSM and check over time if they need a greater clearance. But, bapineuzumab and Gant, they also see plaques and oligomeric A beta. What makes aducanumab different? What underlies its success?
Well, I don't exactly know what the bapineuzumab and the solanezumab data really mean. We didn't really compare them head to head, so it's not really fair to do a comparison of 3 different studies. However, we can say the following things. First of all, aducanumab does not bind to free A beta in the blood. So it stays with a long half-life, about 3 weeks, unbound in the blood. Then it during that time migrates over the blood-brain barrier about 1.5%, where it then will bind to amyloid, plaques, fibrils, and soluble oligomers, and get stabilized for a long time. So over time, you increase the drug levels significantly, and then the removal process starts. With bapineuzumab and also solanezumab, there's already binding of soluble A beta in the blood, so we don't exactly know, what that means. Does that mean that there is still enough drug, and maybe the Lilly scientists can answer that question, is there still enough unbound antibody that can still bind to pathologically relevant species? I guess those data should be
and before we move on to neuroinflammation, is it fair to say solanezumab looks like it's safer in higher doses because of it's not binding to amyloid, it's not causing vasogenic edema or swelling or hemorrhage. And you have some issues in ApoE4 carriers with Ajik Canyon, even though it's the better amyloid clearing antibody. But I'm just wondering, looking toward the future, we were waiting for Risa's results on A4 and the other similar trials. But given that you can use high doses of solanezumab, you saw a 15% improvement using one of the readouts with a significant p-value.
Yep.
I mean, it wasn't exactly a complete failure. I mean, the newspapers like binary events and just say, oh, it failed. But I mean, I was encouraged that even in your major readout, the p-value is 0.09. I thought you guys kind of, admirable that you didn't spin it, but you actually had a better result than maybe what the newspapers were saying when you look at the details. Is it possible that you can up the dose, since it's safe, at a higher level for treating patients early on in the future?
That's a discussion that we've had many times, as you can imagine, throughout the development of so, and I won't revisit the reasons for the decision on the dose. It's fair to say we are considering increasing the dose in studies that are running for solanezumab at the moment, but that decision still has to be taken.
Right. And finally, Roger, on Ajik Canyon, what is the plan now for dealing with E4 positive and negative patients in terms of avoiding adverse events? Well,
as you know, the only really important adverse event that was seen so far was a phenomenon called ARIA, amyloid-related imaging abnormality. In 2/3 of the cases, it's non-clinical, so it wouldn't be even detected in a clinical setting. It was detected by MR safety scans. And then in the 1/3 where it was clinical, it resolved. Mostly they were mild to moderate headaches. So upon further analysis of this, the thinking now is, maybe this is mechanism related. Maybe the removal and also the fact that it occurs only in the beginning, the first 4, 5, 6 months of the treatment and never ever reoccurs. Even now, the first patients, I think, are now entering the 5th year of treatment without ever having ARIA reoccurring in later stages. So I don't even know if ARIA is a side effect or more like a pharmacodynamic readout. But to come back to your question, the effect was clearly dose dependent. The higher the dose, the higher the frequency. And interestingly, it was also ApoE4 dependent.
Right.
So in the ApoE4 carriers, there was much more than in ApoE3. So therefore, the decision was made to dose with the placebo and the low dose and the high dose in both ApoE3 and ApoE4 groups. And the ApoE4 carriers are dosed on the lower dosing scheme at this point in time. But we'll have to see if the safety continues to look good. Then there's a possibility that will also move up to the higher dosing.
So, Hugh, neuroinflammation, big part of the disease, and again, the resilient brains say that if we can stop neuroinflammation, we can deal with quite a few plaques and tangles. Recently, just over the last several years, genes like CD33, Trem2, a slew of other innate immune genes have popped up as probably the most dominant new subset of genes in Alzheimer's disease. What are our prospects for treating neuroinflammation, and when do we do that? We have to treat amyloid early. We probably have to treat even tangles pretty early. Neuroinflammation, I know that's what patients with symptoms probably need the most. Their brain's under attack. But do you treat neuroinflammation early, middle, late, all of the above, and how do we do it?
So I suspect everybody knows now, microglia are now the answer to every known neurological and psychiatric disease. So it's a bit like epigenetics, don't quite have enough information to be sure. So the problem with the microglia biology is we don't know enough about the human condition. And we heard a discussion earlier about the value of transgenic mice. There is clearly a real need to study the human brain, the molecular pathology of the human brain, and to get an insight into when these microglia first respond in the brain, how their activation states then change as the disease progresses. And I think one of the exciting things, in fact, for a person who's worked on microglia biology for so long, the really exciting things is this cloud of innate immune genes that has appeared in the GWAS studies. And of course, what one has to remember is these innate immune genes are not just expressed above the neck. You know, there is another bit attached to the neck. And old brains are attached to old bodies, and the innate immune system systemically is changing too. So if we want to understand what the microglia are doing, we need to understand the background of the patient, the comorbidities. We need to do the pathology on people who've been studied for some period of time. And this is where studies like the Dementia Platform UK, combining with mechanistic studies, are real opportunities to gain insights into this microglia biology. My own prejudice and the prejudice of the lab where we do manipulations of the microglia biology is that too many activated microglia is not a good thing, period. Someone else uses that word period all the time. I think it's in the White House somewhere, but period. Anyway, it's very profound. If you put period after it, it means a lot, okay? So the important point here, however, is we need to gain insights into these cells, and if there are too many, can we find ways of modulating their activity? But we're also going to modulate other innate immune cells systemically. So it's the two things together, how they go hand in hand. And the trade-off between interfering with potential homeostatic functions of these cells versus their pathological components. But I think we need to join up with immunologists. There are, quite frankly, not enough immunologists involved in neurodegenerative disease research, and there are huge opportunities. We know an enormous amount about the innate immune system. And I know Christian will speak to CD33, Trem2, some of the specific molecules. But I think the opportunity that lies in seeing this cloud of innate immune molecules that are in some way regulating the activation states of our innate immune system, this is a real opportunity.
So Roberto asked a question. Amyloid removal requires microglial activation, but their own activation is linked to neuroinflammation and pathology. How do we reconcile these? So maybe you can talk about different types of activation.
Okay, so the whole problem here now is that we can find a paper to support almost any point of view you like. So it is abundantly clear that microglia will phagocytose A beta and will phagocytose plaques if you encourage them with help from an antibody. But on the other hand, if you take the microglia away, at least in animal models, it appears that it has rather small impact on both soluble A beta and plaques. And yet, if we believe what we see in the animal models, the neuropathology is in some way ameliorated. So activated microglia can remove the plaques and the soluble A beta. There's a gap in our knowledge. Precisely how much do they remove? Yeah. Thinking of the other routes by which the A beta gets out of the brain, drainage along the perivascular spaces, transport across the endothelial cells. Actually, it's all qualitative. We really don't have the sort of quantitative measures that we need. So I think this idea of being aware that there are trade-offs to be made, that activation and the secretome of the microglia has the potential to interfere with synaptic signaling in multiple different ways. At the same time, we want to get these cells to play their role if it's necessary, I think it's a caveat, in the removal of the A beta.
So you mentioned the body, the rest of us below the brain.
Yeah.
Is there a role for peripheral inflammation in AD? Do we have any evidence of that?
So we know from animal models, all their strengths and weaknesses, there seems to be evidence that systemic inflammation can exacerbate disease. In our own studies, I've done in collaboration with Clive Holmes, if you follow people over time, it is clear that people with systemic inflammation do less well over a period than people who don't. The extreme of it that everybody knows who's a clinician is delirium, that systemic inflammation can exacerbate symptoms way over the top to points of cognitive impairments, hallucinations, attention deficits, and so on. So I think we do know that systemic inflammation can impact on the diseased brain. And I think one of the interesting challenges ahead is to tie the epidemiology to the genetics. So epidemiology of subjects with systemic inflammatory disease, can we tie that to the genetics that have come out of the GWAS studies? And I think there's real opportunities there. And
Christian, you've done some work on Trem2 in particular. and especially on the secreted TREM2. And TREM2, can you tell us a little bit about TREM2 and STREM2 as a biomarker? And there's some debate about whether TREM2 is a good guy or a bad guy. CD33, we know if we turn it off, microglial cells eat more A beta. TREM2, if we turn it off, it seems it's the opposite. So what's your feeling?
Well, I think it's very easy, period. Basically, the genetics is going to tell you. We have a lot of mutations in TREM2. These mutations are loss-of-function mutations. So obviously, we have to bring back TREM function in these microglia. Now, what's very interesting is if you take out TREM, the microglia is locked in a homeostatic stage. So if you would cross a TREM2 knockout with an APP-PS1 Alzheimer's mouse model, they fail to be induced. So they don't show the neurodegenerative disease signature anymore. There's no chemotaxis. There's basically no phagocytosis, and so on. So obviously, some kind of activation, at least during a limited amount of time, is something really, really positive and probably also a therapeutic target. But of course, this cannot last forever. Now, we looked in human patients, so we went into the Diane cohort with the inherited patients, and what we found was that soluble TREM2 goes up at -7 years before expected year of onset. So that seems to be, to my opinion, a response to some neuronal insults. And when you then look what it correlates to tau, but not to amyloid. So that means it may correlate to neuronal injury and not the buildup of amyloid plaque. So it comes later, but there's a response to neuronal injury. So I think microglia may be very much important to remove apoptotic debris in the brain. And that's probably also the reason why we have mutations linked to many different types of neurodegenerative disorders.
Well, TREM2, I mean, the homozygotes for the R47H mutation have their own form of dementia, the Nasu-Hakola, that begins with inflammation. So we talk about amyloid and tangles and inflammation following, but there's a clear case where inflammation is probably coming first, and then that's leading to tangles. And then you have heterozygotes for R47H, which is very infrequent, less than 1%. But I think even the last meta-analysis says if you carry one R47H allele, you have an increased risk of over twofold. And there you're seeing classic Alzheimer's disease with plaques and tangles. Yeah. So how do you put it together? Does that mean that inflammation can also drive that wheel toward plaques and tangles, not just the other way around?
That's very hard to tell. I think it's one of the biggest questions at all in the entire field. So why are mutations in one and the same gene causing different types of disorders? We have no clue about it. The R47H mutation causes Alzheimer's disease. The T66M mutation causes FTD-like syndrome. Both of them may be loss of functions. T66M is like a knockout, basically. R47H may affect stability of the protein, so maybe a bit more subtle. Maybe that's the difference, but we really don't know yet.
And the big question is, how are we going to treat neuroinflammation? How are we going to actually get a handle on it? And of course, the amyloid hypothesis and the amyloid-related genes have been around for 30 years now. We've only gotten a handle on the innate immune genes like CD33, TREM2 just over the last several years, so they have some catching up to do. But maybe Michael can tell us a little bit. Michael and I go way back decades. We worked on Huntington's disease together back in the early 80s at Mass General. And you're still working on Huntington's disease, and you're on a panel on Alzheimer's. So tell us how that happens.
Well, I just want to say it's wonderful to be part of a panel. Rudy hasn't changed at all since the early 80s. I've lost a little hair, worked a little harder. And then the other thing I want to say
My belly's bigger.
Yeah, or maybe. And Marty Samuels spoke about him being not memorable. And I just want to say, him up there with his clinical, beautiful acumen and sensitivity. I still remember him doing the rounds at the Brigham and Women's in the mid-'80s with the same sensitivity. And then the other thing I would just say, when I was at the Brigham and Women's and Children's, Mass General was very far away. There was a big chiasm. And that new partners organization, which is doing so much for me, is a great model about what collaboration, cooperation, and working together can do, which is so needed in this field. Now, let me say, I've been at Teva, 4 to 5 years now. And when I arrived, Teva was committed to multiple sclerosis with Copaxone. But I was keen to take Teva into other neurological and neurodegenerative disorders. And the key for us was really thinking about convergent pathogenic pathways and having shared targets for drug development. And clearly, when we looked at multiple sclerosis, disturbances of both innate and adaptive immunity, with a whole host of other pathways, mitochondrial dysfunction, various other pathways that were also shared with a whole host of other neurodegenerative disorders. Huntington's disease came to the fore because of my past experience, but also Huntington's disease represented a very unique model disease. Dreadful disease, frequency underappreciated, today recognized 1 in 6,000. When you look at the people who carry the mutation, it's about another 4 to 5. So the frequency is quite similar when you include the pre-symptomatic population, destined to be ill but not yet ill, about 300 to 400,000, Europe and North America. And then, of course, with Huntington disease, you have absolute single mutation. You can predict who's going to get the disease, and that's taught us a lot. And then, of course, when you look further, there are cohort studies. We know how to determine progression of the illness in 1 year, so we have endpoints. So this represented an important entry for Teva into this. Today, Teva has now committed really hundreds of millions of dollars to research in HD. We have a PDUFA date for a drug just April the 3rd coming, who knows, but then also in phase 3 and phase 2 development. What we learned from a drug in multiple sclerosis, which was laquinimod, was that this is a drug that appeared to have major neuroprotective effects greater than its impact on relapses. So it had a disproportionate effect on neuroprotection and protect against atrophy. And the challenge became, what is the target for that particular drug? And through a whole host of cellular, other assays, eventually RNA-seq analysis, the aryl hydrocarbon receptor was determined to be the target of laquinimod. And this is now recognized to be an important target for neuroinflammation, moderates both astrocytic and microglial function, decreases both chemokine and cytokine release, and has big impact on the disease. So with that information, we went to look at other models and in fact looked at Huntington's, other models, in vitro, in vivo, iPS cells, and recognized that both in Huntington disease and in MS, there's disturbances in innate and adaptive immunity. You look at the people who are destined to be ill, they have microglia activation with a collaboration with the group that was here this morning with microglia activation 10 years prior to onset. And this gave us an idea that certainly treating patients with Huntington disease, animal models showed rescue of both motor and striatal atrophy. And so we began a study on Huntington disease in phase 2 development. And I would say as we look at this, what we're most interested are those drugs that we call hub molecules. In other words, they act on shared pathways and can influence these pathways in multiple ways. And both multiple sclerosis and Huntington disease represent initial model disease from which we'll learn a lot and then may be able to embrace larger neurodegenerative disease like Alzheimer's disease. And so we're, of course, looking at other pathways, pathways of, apoptotic pathways, caspase-6 activation, which are shared both between Alzheimer's and Huntington disease, other pathways of synaptic dysfunction, defects in both synaptic and extrasynaptic dysfunction. One of the only drugs approved for Alzheimer's is memantine, which really has impact on extrasynaptic NR2B receptors. So we're looking at all of these, but using Huntington's and multiple sclerosis as a model disease from which we can learn a lot in early phase 2 proof of concept studies before we embark on major, bigger neurodegenerative disease. Those on their own are big enough, but certainly we haven't given up on this, but we're learning more at this stage. The other thing I would say, when I listen as an outsider to the discussion, for me it's about timing of treatment. And the problem is, of course, with Alzheimer's, again, accurately predicting who's destined to be ill. And certainly when we talk to the regulators, we couldn't do a trial right now, even in Huntington disease, in pre-symptomatic treatment, before we've shown in early disease that we have some effect. So we're also going to have to look to changes in the whole regulatory environment to embrace the upcoming tsunami of neurodegeneration that's on us in an effort to really progress this field.
So in terms of targets, you're working on the sigma-1 receptor.
Yeah.
Tell us a little bit about how you think that works. What's the prospects for targeting sigma-1 in Alzheimer's or in other neurodegenerative diseases?
Just to say, this was a study done about 8 years ago, and I happened to meet the patients who were telling me how much better they were feeling. And predopity in this drug only bound, it was meant to be a D2 receptor antagonist, had very low affinity. We brought the drug in, determined very quickly that this had major affinity for the sigma-1 receptor. And we just concluded a phase 2 study in Huntington disease that surprised us completely. Because what we were able to show, early study, proof of concept, not fully powered, but still significant results, that we could show slow the functional decline for the first time using an endpoint that had been used in 13 trials before, which had never shown any movement, about a 50% decline in this particular endpoint. And now we're working with the end of phase 2 meeting with the FDA to design the next trial. But when we look at this, and we look at what it does, and we look particularly at some of the effects of sigma-1 receptor activation, one of the big effects is increasing BDNF. And we know that BDNF is reduced in Alzheimer's, also reduced in a whole lot of other neurodegenerative disease. We know that certainly in vitro you can protect against A-beta toxicity and various forms of toxicity with BDNF. And so this just raises questions for us about whether some of the mechanisms that are underlying potentially some of the neuroprotective effect of the sigma-1 receptor activation may also be operative in other diseases like Alzheimer's disease.
It's something, I know there are quite a few groups now targeting sigma-1 in Alzheimer's. So back to this question of stage and timing. I mean, you know, precision medicine, we talk about right patient, right drug, but it really is right patient, right drug, right time. And there's a question that came from the audience. How does stage of disease come into play for treatment with anti-amyloid therapy? Is symptomatic disease too late for a BACE inhibitor? And I would extend that to say, are we diagnosing Alzheimer's disease too late? I mean, imagine if in cancer, we didn't diagnose it until there were symptoms, that somebody had to have organ failure in a 1-inch or 2-inch tumor, and then you treat with a tumor suppressor. Or in heart disease, imagine if we treated only after you got a heart attack or congestive heart failure. Is it kind of absurd that we wait till the brain is degenerating and there are symptoms, and then we try to treat the potential cause based on genetics by treating amyloid? And along with that, someone asked, would there ever be a trial outcome, and this is for you and Roger, that would convince you that amyloid is not a useful target?
Rudy, let me just start off with the first question. Just think about heart disease, and we were involved in a lot of those trials. You could only go to primary prevention after you've shown that it was useful in secondary prevention. So this concept of the statin and that kind of protection, if we really want to treat very early, and we might need to, we're going to have to change the whole paradigm, and the statin treatment paradigm may not be totally applicable.
Yeah, I mean, I was going to come on and say, actually, on the point about early diagnosis, I mean, obviously, we all want a disease-modifying therapeutic, but right now, even if we had one, the healthcare systems around the world are just not really set up to deal with disease modification. And one of the key gaps, of course, is in this question of diagnosis, and early diagnosis in particular. I mean, the proportions are still controversial, but it's clear that far too many patients are being diagnosed late in the course of the disease from moderate stages onwards, and many, of course, are never receiving a diagnosis. And that's a key issue that we have to resolve. There's a number of ways we'll address that over time. Partly, of course, it's through education of patients, removing the stigma and the fear of the disease. And then, of course, also education of doctors, particularly the primary care physicians. Beyond that, I think there's a number of other things. Of course, I think the amyloid diagnostics will be an important player in moving earlier in the disease, and their availability, be it PET or CSF beta, I think will be very important. We also clearly have a gap in that what we lack right now is a non-invasive, simple biomarker, simple, cheap, rapid biomarker that we can use to identify patients who have amyloid in their brains or are at risk of developing Alzheimer's disease. And that's something else we're going to need if we're going to have, we're going to move to the point where we can effectively identify patients and then treat them early in the disease, whether they're coming in with a subjective memory complaint to a primary care physician, or whether we end up catching it through a routine health screening check, which I think is where everybody would want us to go eventually.
Roger?
Well, we know from several studies that the biology of antibody-mediated amyloid removal works at every stage of Alzheimer's disease, very early, preclinical, and also late-stage disease. So the question is how long...
What do you mean by works? What do you mean by that?
Well, you can remove amyloid even in late-stage Alzheimer's.
Okay, but does it do any good?
But the question is, how early do you need to do that in order to preserve neuron function? And studies have shown that the beginning of the pathology starts 30 years before the onset of the first clinical symptoms. So we have that long preclinical time in which a diagnosis can be made and a therapy can be done. So from this data, it is very clear that we need to go early. We need to identify people at risk early. It certainly cannot be done with PET. PET is a wonderful tool for research and for development, and I think was key to the success of this, trials that are looking good at this point. But PET will not be a useful tool for screening. So we need like a $1 blood test, a simple test.
It won't be useful because it's too expensive, or
well, it is, first of all, it is not available. Secondly, when in Switzerland, our site in Zurich, is probably one of the areas with the highest PET densities on the planet. Yet when we would use all of that capacity 24/7 for Alzheimer's screening, we would just have 1% of the demand, and that's a PET, haven. So the PET machines are not there. The radioactivity is difficult in terms of logistics, low shelf life. So you have good transportation practice to bring the radioactivity and so forth. So PET is a wonderful tool for research, but it's not going to be a marker for screening. So work really needs to be done to identify, hopefully, a blood marker that is meaningful to get at these populations of brain amyloidosis patients. And then let me add one more thing. There's not only amyloid. There's also tau amyloidosis, TDP-43, C9orf, and so forth. So in the future, we're going to need a toolbox to identify patients with those protein aggregating diseases before the clinical onset, and a match toolbox to remove those protein aggregates. And I think together that will then finally end up in a personal type of diagnosis and treatment, and maybe even primary prevention.
So along those lines, there was a question. What else do microglia remove in the brain? Do they remove these other pathologies as well? Could we use microglia to do that?
So before I answer that question, let me say, we heard earlier from the very first talk of the morning that there are 47 million people worldwide who have this disease. So desirable as PET may be, they're not going to see it. Desirable as a $1 test may be, they're not going to see that either. There are lots of people around the planet working on ever better psychological assessment tools, and I think this world of apps and clever people working on sensory motor function and other psychological domains, in the same way that psychiatric disease has moved forward, taking a domains approach, domains attached to different regions of the brain, functions of the brain. I think we shouldn't forget, the output of the brain is behavior, and we should get better and better at assessing behavior as ways in. And I think it can be as sensitive as some of the other things we've heard about. Do microglia do something beneficial in the brain other than remove amyloid? Well, of course, this is an ongoing discussion about the role of the microglia in brain homeostasis, in brain plasticity, and so forth. It's pretty clear the simple answer to that, like so many other things, is actually we don't know. But there are people who are concerned that maybe the microglia really are involved in this homeostatic function, but there are other reasons to believe that they, again, might be minor players, and it's assessing that balance, because if we want to take the microglia away for benefit, we don't want to disturb this homeostasis. But I'll leave it at that.
So we're just about out of time, but 10 seconds each. 5 years from now, 10 years from now, are we preventing onset of Alzheimer's disease through screening and treatment?
I think we'll have disease-modifying therapeutics. The potential to prevent will be there, but we still will have some work to do on the healthcare infrastructure, I suspect.
Roger.
5 years from now, we'll have a drug that removes amyloid, and 10 years from now, we'll have a combination therapy with this drug and a tau or a BACE inhibitor.
Hugh?
I think there are disease-modifying opportunities, and they involve modifying the systemic diseases associated with the progression of Alzheimer's disease.
I think in 5 years from now, we are close to combination therapy, which really prevents and removes amyloid.
And I remain optimistic like the group, but I think some of the insights, we've got to do much better to break down the barriers between these neurodegenerative diseases and pool knowledge, pool know-how, and be able to do quicker clinical trials in an effort to push the field forward.
Great. Well, thanks to all of you. Thank you.